Welcome to the Organic Compounds lecture for your biology honors course. You might notice that the title for this lecture says supercarbon, and that's because carbon is what makes organic compounds. Organic compounds are really a long chain of carbons that can be folded up into rings or different shapes, and then also added a different functional group to give them different purposes. Carbon is what makes up most life forms, which is why they're called carbon-based life forms. And there's 4 reasons why you should recognize that carbon is amazing. First, it's abundant. There's tons of it everywhere. It has 4 valence electrons, so it can bond with 4 other atoms at the same time covalently. It's versatile, so it can bond with a lot of different things, and it's also very stable. So once it's bonded, things don't fall apart. You should be using the Organic Compounds Note Guide provided to you in Canvas. Please fill that in as we go through this lecture. So organic compounds are critical to the structure and function of living things. All living things are made up into 4 categories of organic compounds. So we have the carbohydrates, things like sugars and starches, proteins, usually found in meats, nucleic acids, which is DNA and RNA, and lipids. Any organic compound can fall into one of those 4 categories. They're all made out of different monomers, which are smaller structures that bond together to produce polymers. So if we are trying to get these long chains of carbon to have different jobs, what we do is we add what's called a functional group onto that organic compound there. So if we have a long chain of carbon, we sometimes call that the carbon backbone. We can add a functional group like a phosphate, and now that's a nucleic acid. Or something else that will make it into a different compound, like an OH group makes it a carbohydrate, an NH2 makes it a protein. This long carbon double bonded to an O and then an OH makes it a lipid, and then nucleic acids is a phosphate group. This just determines what function that long chain of carbons is going to have, and that's why it's called a functional group. So we're going to start with carbohydrates, and carbohydrates, just like all of the other organic compounds, is a large compound or a polymer made up of a monomer. So these are used in the body for energy and for structure. So in our bodies, we break down carbohydrates and we use them in mitochondria to make ATP energy. For other organisms, they might use them for structure, like plants build most of their cell wall is out of carbohydrates, cellulose. The functional group for these is OH, and that's why we have all these little OHs hanging off over here. Most of the time, we see them into a ring shape with some OH things hanging off of it. The monomer for these is a saccharide, and saccharides are simple sugars like this glucose here. See how it only has one ring? That's the a monosaccharide, or fructose here. But we can also combine carbohydrates from monosaccharides like glucose, where there's only one ring, into disaccharides like sucrose that might have 2 rings. So instead of like the previous diagram, it would have more of a 2-ring structure like this. Maybe attached to each other like that, something along those lines. A polysaccharide would have many rings, and those we also call complex carbohydrates. So that's things like starch. All of these fall into the category of carbohydrates. Some other things that you might want to reference about carbohydrates is that they only contain these 3 elements: carbon, hydrogen, and oxygen. When you look at the diagrams here, you can see that they're all only those 3 elements. These carbohydrates provide fast energy. Fast energy, meaning that when we break them down, we get ATP from them really quickly. Some of these other compounds are also used for energy, but carbohydrates is the fastest for energy. Our second type of organic compound is called a protein. And proteins, again, are large compounds. They're also polymers. They're used in the body to form tissues and as cell membrane gates. We have a lot of proteins in the cell membrane. And you'll learn about those later and all of the jobs that they do there. The functional group for this one is NH2. So anytime you see that on a diagram, you're going to know that that's going to be a protein. The monomers are amino acids. So amino acids are small little groups of carbon and NH2 that then bond together using peptide bonds to make a long protein. So if we have an amino acid here and an amino acid here and an amino acid here, and we bond them all together, then that is going to give us a protein. Okay. We see these a lot in meats and fish, but also we see them in legumes, things like soybeans and peanuts. So there's lots of different ways that you can get protein in your diet, but it's very important to do so. One thing I'd like to note is that proteins do not provide energy for cells, except for as a very last resort. So proteins are not going to be used as energy. Many times students think that they are because they think about high protein shakes and things like that for people who work out, but that's people that are trying to build muscle, trying to build meat on their bones, not trying to get extra energy for doing their activities. Carbo loading, carbohydrates is what you do in order to get extra energy. So remember that proteins are for forming tissues and building structures and cell membranes, not for energy. Okay? So this is something that we have talked about in the previous chemistry unit. So try and answer these questions on your own, and then we'll go through those answers. What is another name for a biological catalyst, and what do they do? If you were to put one into a chemical reaction or a chemical equation, what would that look like? Pause the video here to try and answer these questions on your own before seeing the actual answers. A biological catalyst is called an enzyme. And the point of a catalyst is to reduce the activation energy and allow a reaction to occur more quickly or allow it to start. So anytime that we have something that's going to be happening in a biological equation, an enzyme is likely used to start that reaction and allow it to happen. However, the enzymes themselves are not changed in a chemical reaction. So what we'll have is a substrate, which is what reacts with the enzyme, and the enzyme then allows that equation to occur, and that produces the product. But the enzyme is not changed, so we'll also have that enzyme left over at the end that allows that product to be made, but doesn't actually get changed itself. Okay. So just think about this. Do you think it would be easier or more likely for your body to use one enzyme for a reaction more than once? Meaning, are we going to use the same enzyme over and over again, or are we going to use a new enzyme every time we need to run that reaction? In addition to that, do you think it's important for your body to be able to start and stop chemical reactions at certain times throughout your life? Or are things just going to go all willy-nilly and be able to happen at unpredictable rates? Well, as I'm sure you guessed, your body definitely wants to regulate things like that. So what your body can do to regulate it is by changing the active site on the enzyme. So enzymes are very efficient because one enzyme can run the same reaction over and over and over again. Since the enzyme isn't changed, we don't need to make a new enzyme to run the same reaction. However, an enzyme is very specialized for a very specific reaction. So we don't have one enzyme that runs a lot of different reactions. We have one enzyme that runs the same reaction over and over and over again. So here on your screen, you see that you have an enzyme and that it fits with a substrate. We call that method the lock and key. That ensures that only the particular enzyme that is needed is fixing only the substrate that needs to be changed in a chemical reaction. So if we don't want that substrate to be changed at that time, if our body needs to regulate it, we can do a couple of different things. One thing that we can do is put a little cap inside the enzyme and change it there. So now the substrate will no longer fit inside of that active site and the reaction won't take place. And then whenever want to have that happen, we can just get rid of that and now the reaction can take place again. Another thing that we can do is change the shape of the active site by having something else attach over here. So if we have a little blocker that attaches to this enzyme, then that might slightly change the shape of the active site, and again, the substrate won't fit in there. So that's two ways that you can stop a reaction from occurring, by either blocking the active site or changing the shape of the active site. You should add that to your notes under proteins because you'll see that again later. One important thing to know about proteins is that if they have an exposure to a pH change or a temperature change, they can do what we call denature. And denaturing an enzyme means that you break all of the bonds that are holding it in a particular shape, and it just becomes a long worm of amino acids. And if the protein is not in its particular shape, then it's not going to be able to do its job. So destroying the protein shape disrupts its function. Normally, it would be all folded up with hydrogen bonds holding these things together. Since the denaturation occurs with the pH or temperature change, that makes the protein unwind and it no longer works properly. This occurs with enzymes or any other protein. That's what happens when you cook your meat. You denature the proteins that are in there, and that's why you see the color change and the texture change of cooked meat. Right. Nucleic acids are the third type of organic compound that you need to know. Just like all of the others, there is a large compound called a polymer made up of many smaller pieces called monomers. The monomer for this group is a nucleotide, and a nucleotide consists of three main parts that you should know as well. So there is a sugar, either deoxyribose or ribose, depending on if it's DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. And that also has a phosphate group attached to it. And then those will alternate going down. This would be a sugar, deoxyribose, phosphate. And then we also have one of four bases attached to the sugars. So the bases, which you have probably learned in the past, are thymine, adenine, guanine, cytosine, or uracil, are attached to the sugars. Those are the bases right there in the middle. And then the backbone is alternating sugar and phosphate groups. Okay? So the phosphate here is our functional group for nucleic acids, and that's what gives it its function. Our last organic compound is lipids. And again, they're a large polymer. These are used in the body primarily for insulation, and that's what our cell membranes are made up of. They're also used for energy, but this is usually slow energy. This is how we store our energy in animals. Plants store their energy as carbohydrates, and animals store their energy as fat. The monomers are 3 fatty acids and a glycerol, and the functional group is this wacky thing right here. You'll see that attached to the carbon chain. These are all hydrophobic or non-polar, which means that they do not get along with water at all. Okay, so that's all our oils and fats and things like that. One interesting thing is that we can modify these guys. Okay, so this kind of references the cartoon at the beginning. This is a fat. He realized that he's hydrophobic. Sad day. Okay. So one thing we can do to make a lipid partially hydrophilic is we take off one of the fatty acids and add a phosphate group, right? See, here's our phosphate group, and it's attached to some other stuff, but this makes a phospholipid, and that's what all of our cell membranes are made of. So this is going to be a really important compound for our class once we get into cell membrane structure. Also, anytime that we see things in a ring shape, we'll call those sterols, and that's things like cholesterol and steroids. It's just a fat that is in the shape of a ring instead of being in a long line like these guys are right here. Okay? Please make sure that you use the notes that you took during this lecture to answer any questions on your labs and assignments and prepare for your tests. Please send your instructor any and all questions that you might have over this topic. Thank you so much and have a wonderful day.